So this is supposed to be a brainstorming session. That means you all need to contribute your brains to this session. And so the main theme is about, what can we do to improve classification? How can we contribute to simplifying the process? So just as a sort of a recap of what we've heard today, there are 4 points I thought we could touch on today. So one thing is, should we be developing a common scheme for classification across virus families? And for that, we have to think of what is the purpose of classification, right? So we know it's to better communicate with various stakeholders. And so we have to keep in mind the different needs of the different stakeholder communities. And what we need to figure out is whether we can combine these needs with a common scheme. And maybe it's not possible. So for this, I'm thinking, how the example that Betty was giving us of SARS-CoV-2, I think is a good one, where we had the Greek letters to better communicate mostly with the outside, but it was, with the public, but it was also useful for research purposes, but it's a very broad classification, right? And then we have a less broad classification, and we can take the Nextclade example, and then the Pango that is very detailed and sometimes too detailed to facilitate things. And then I thought it was interesting when we heard about the genotype-based classification, like the rotavirus, where they're using purely nucleotide level, the 80% cutoff that you were talking about. And then we heard for the adenovirus, the sequence typing, they do it by sequence typing. So there's no more of the using serotyping for classification. But Tavis, I thought, described it really well when he said we have to think of this as multiple dimensions, where for some virus families, you may need more than 2 criteria in your schema, but some you will need far more. So if there was a scheme, a classification scheme that could be flexible enough where you can enter your different components or your dimensionalities as modules and develop such algorithm. And what we would need to determine as a group is what are the criteria for this algorithm. And we saw, was it you, Elliot, that was showing the algorithm when you go through the different, for your pox virus?
The demarcation criteria that's used for the official taxonomic classification.
Okay, yeah. What I thought was interesting was the filovirus example, where the naming scheme was interesting because they have that shortened name, that the abbreviated classification, then you have that medium length that has a little bit more granularity, and then you can go to the full length name. And I thought that was something to keep in mind. And the fourth point is who should be responsible for keeping up with classification. And in my mind, I keep on thinking about, Liliana and I had a discussion about SARS-CoV-2, where right now it's sort of Betty that's the translator between, the Pango, the different lineages, and then communicating to the different stakeholders, the in vitro people that do in vitro work, the in vivo people. But, if there's no Betty, what do we do, right? We need an AI Betty, basically. So that's where I wanted to start off the group. And so, Elliot, what do you think?
Well, so to start off, these are definitely the big questions that we set out wanting to ask and answer to the extent possible when we were designing this whole workshop. And we will hear more about each one of these topics tomorrow and Wednesday. But now's a great time to start off the discussion, because we've been presenting with the different subspecies classification schemes of very different virus families. So we sort of had the big picture at that point. And we have a bit of the picture now, a little bit of the picture as to who the different stakeholders are, though I don't think we yet have a full appreciation of each different stakeholder and what their particular needs are. And I hope we'll hear more about that again in the next 2 days. As I think about pox viruses and what about pox viruses relates to SARS-CoV-2 and what aspects that are most important that go into the classification scheme. And one of the things I guess we heard about today was the combination of genotype and phenotype, and how genotype may give you the basic phylogenetic classifications in many, though we heard not all instances. We recognize what's important about that phylogeny based upon the phenotype. That is, we still need the phenotypic characters to tell us which lineages are the lineages that we want to follow. And that's certainly true in pox viruses, as it is, I think, in every other virus we talked about. So maybe that's one common theme across all the viral families, is that you need those multiple criteria, which is something the ICTV has been using since its inception. So,
Justin?
Justin Ball, University of Georgia. I think the flexibility in the system has to be there as well, right? Like if you have one of the problems that I think we've done with high path avian influenza is we've decided we had to label every single thing, right? That we found, where I think Tavis showed that they had a group of viruses that weren't labeled. Then when they had that additional information, whether it's phenotype or an epidemiological virus, that if you can have space where you can create something new, you don't have to create a system that constrains you, that you can't actually put in the phenotype in there when you have it. And I think that's a problem with the H5 system right now.
One of the issues, though, is that the genotype often points to which ones should be phenotyped, because it gives you good reason to test something. So it's kind of a little bit circular, that information. And I think it's important to remember that. And in that case, it was when a human got it that they, that then they looked. But we don't always have when a human gets it as a criteria.
Yeah, Virginia here. So along the same lines, so often you kind of, well, at least in the seasonal flu, sometimes you suspect a phenotype, but for some reason or another, the assays you have, ferret antisera in this case, don't pick it up. So with the pandemic H1s, for many, many years, there was clear sort of sweeps of very genotypically defined variants that seem to have made it different in humans. Otherwise, they wouldn't have replaced the previous genotype so fast. But ferret antisera just didn't see anything. And there, yeah. So if you lack the appropriate assay, so you need to be in a position to divide things just purely by genotype.
I think this extends on the last two comments, is that positive selection is kind of an abstraction that links the genotype and the phenotype. And so if you see evidence of positive selection, you don't necessarily have to know why it is, but that just indicates that that's an important characteristic of that genotype.
Yes.
I'll come back to this tomorrow in my talk on taxonomy, but I just thought it might be important to be aware that we maybe talk about more than one thing. So a lot of the talks today were sort of groveling around at the bottom of the tree in sort of molecular epidemiology. Whereas actually, there are, huge sort of issues around genotypes, serotypes, and so on, much higher up in sort of this much higher up in the tree. So, for example, hepatitis C genotypes differ from each other by 30%. So they're not the sort of things that evolved since 2020. So, in fact, for it was about 500,000 years ago. So, so their classification and the issues surrounding those are quite different from this sort of dynamic nomenclature that's developing for SARS-CoV-2 and other viruses that are emerging. So, I think it's important to try and make a distinction between level, what level you're working at, and I'll come back to this tomorrow.
Anya O'Toole, University of Edinburgh. I think kind of fitting in with that and also touching on the flexibility point, when you're talking about genotype and phenotype, if you've got, I suppose, different questions, the phenotype may or may not be as important. So if we kind of rewind back to early in the pandemic, a lot of, for us working in the UK, the government was really concerned with number of introductions. And there's not necessarily a phenotypic difference between different introductions, but we were kind of tasked with kind of keeping track of those. So an introduction would have been a different lineage, but it was
a question of timing. Also, you're dealing with an epidemic, right?
Yeah.
I think also a use case here. So the difference between contact tracing versus worried about viruses that are emerging that are resistant to antivirals versus trying to characterize 3 years worth of evolution within a virus that you can say this was the 2020 virus and here's the 2034 virus. And I think that sort of layers upon layers. Yeah, being able to speak to the various use cases.
Yeah, Richard Sherman, National Library of Medicine. I think this is where your dimensionality comes into play. Is like, can we characterize these viruses based on these different properties? One would be phylogenetic relationship, but then the other are different phenotypic properties and things like that. I think it's an interesting idea. I'm not sure practically how you do that, but that's really what we're after, right? We want to be able to have an understanding of these different virus characteristics. Andrew, University of Virginia. So just to abstract that a little bit further, I think we're talking a lot around two modalities. One is analysis and one is communication. And you can think about separating those a little bit, where you can give an address system to a part of the tree, if you want to think about it that way. And you can use that sort of ubiquitously, and then you can separate that from, to an extent, the naming system that you might use to communicate about the disease. And that might combine addresses, right? If you want to adhere to monophyletic structure, you say, okay, this part of the tree gets a special name, becomes B117 or whatever, or these multiple parts of the tree, if you're dealing with recombination and things like that, then that gets a special name, XBB and so on. It seems like, depending on the analysis that you want to do, you could have different systems, and if you always have the systems be the same in terms of the thing that you're using to communicate and the things you're going to use to do analysis, you might run into problems like, for instance, with Pangolin, you have a social attention function for creating new names, right? That globally, where people decide when, using GitHub pull request, or as long as Cornelius isn't on vacation or anything, create new Pangolin lineages, versus something that ubiquitously treats and references the tree structure, and we can talk about challenges with the tree remaining the same or changing and stuff like that.
So using your analogy, is it as simple as addresses and neighborhoods, or is it something else?
I think it could be. I think there's loads of challenges associated with that like people like Betty can speak to, and Usher and the people that work on Usher can speak to, where the stability of the tree and things like impact the classification boundary between the different names. But if you have, and we talked about a little bit about this earlier, where you could almost treat a single tree structure, if you're just pushing into that tree as a namespace, right? As long as you don't change the tree, you can attach additional things to the tree, and then always have a consistent address system that allows you to do consistent analysis. Maybe, again, coupling, where you almost need to do a join between your name system with that you use to communicate about the viruses and the way that you address into some sort of structure. But yes, to answer your question, yes.
Yeah, so following up on that, for the mpox, monkeypox outbreaks and the tree structure, we've sort of adopted, at least tentatively, this sort of hierarchical system where there's on one level the clades that Elliot talked about earlier, And then for a human outbreak with sustained transmission, we decided to give that a label. And luckily, we only have, or at least we only have established a label for one of them. I'm not actually totally en courant with what happens with more recent things. But then within each of these outbreaks, there is this sort of lineage system, whose purpose is basically just to track sort of, really the epidemiology with individual mutations necessary to label a lineage. So there's really no sense that this is any phenotypic meaning, but that you have sort of a group that goes back to one zoonosis, which has a label, and within that you have another address system if you want.
So I think one of the commonalities I think that we're hearing is we have to start with a good, accurate genotype to the extent possible. Then we start to overlay. So that raises a question of when we start to generate those genotypes, inevitably how we do that is going to differ to a lesser or greater extent, depending upon the virus that we're talking about. So do we use the whole genome? Do we use one gene? Do we use multiple genes? Do we use one segment or multiple segments? So are we going to need to define a system of experts who make the choices for each particular virus and update, modify those choices as necessary, depending on an outbreak, because that could change during an outbreak. There could be unforeseen changes due to enhanced recombination, for example, that you haven't seen before that now has to be taken into account. Elodie is right.
Rachel Cahoon from Edinburgh University. I think that one of the really great things about having a community of people during the pandemic who were helping with the pango lineages was that whilst there were criteria for what they were choosing, they did also influence, and how the virus changed, influence what the criteria were that we were trying to designate on. So early on, we were interested in geographic location, and that evolved over time to other things we were interested in. But I think that having a community of active people who can influence, who aren't just one small team of people who've self-designated for a particular virus, will open it up so that others can have their say about what's actually important to them.
Ryan Connor, NCBI. I think the issue of what would need to be sequenced to support a given scheme, I think ties into points I think Celeste and I think it was Richard made, which is basically a perfect system that no one adopts is not a good system. So you need something people can use. And in the case of sequencing, depending on the people who are most likely to be in a position to do the sequencing for the surveillance, like what technology, what skill set they have access to, I think should be an important factor in deciding what you do whole genome or what genes or whatever, like what skill set, what technology is needed to sequence those things.
Yeah, and I think that just to add on to that, the other thing Celeste mentioned is establishment and maintenance are two different things. So I think what the two of you just kind of implied that when you establish, you have to bring in the right stakeholder group. It can't just be a panel of experts. It has to be a panel of stakeholders. And then you have to figure out how you're going to maintain that system going into the future.
Sean, Minnesota Department of Health. I first of all, I do think we do need to maintain like a group of experts or a group of stakeholders for specific pathogens. And I'm going to bring up the rotavirus B. The reason rotavirus B classification has the whole genome scheme haven't been updated is because Douglas Mathaler, we actually know for years, and he no longer works for the Minnesota Veterinary Diagnostic Lab, and he jumps to industry. So you rely on the whole classification into one person is just, unrealistic. And I also believe that, my PhD was done on Illumina era, so I voted for whole genome rather than single genes. And nowadays, the cost of sequencing one particular gene is basically the same with sequencing a whole genome, from like a high throughput sequencing lab perspective. And Yeah, and one thing I would want to bring up is to bring the quality perspective, because I do work in a clinical lab. We do not want garbage in, garbage out, and I do believe for each particular viruses to have established a panel for any labs that wants to do whole genome sequencing or even single genes, be able to validate what you are sequencing out is absolutely important.
Hi, Andrew Beck, US CDC. I would like for a second to talk about I guess it's the stakeholder and the audience argument for a second, and mostly from the perspective of having mercy on our stakeholders. I think that there's in virology world we have our own shorthand for our special prototype strains, or for the passages that it took to arrive at a particular vaccine attenuation or something like that. And that's the lingua franca to us of our particular viruses that we care about. But there's I think that there's a really great need for clarity and unambiguity when we communicate these things to the stakeholders. And that can include policymakers, that could include the epidemiologists that we deliver the data to, who sometimes have modeling decisions to make and that type of thing. That can include health bodies to whom we're accountable and we may even receive funding from. That can include other geneticists and other researchers, of course. That's who we are in the room here. But I think that there's this idea that we need to judge who is the person who has, I think, the most critical need for unambiguity of the data. And that's the person, aside from the research community, that's the person we really need to think about. So if I have to sit in a room with a group of our epidemiologists and explain the genetic data to them, it needs to come across crystal clear and quickly, without having to give the backstory of where my virus came from and all of the special names we have for all of the strains. So I think there's a place here for the clarity and the unambiguity argument, because ultimately I think that helps the public see the value in what we do as well, because it makes things a little bit more easy to understand for those who don't have to look at this stuff every day. Thanks.
You're saying that public health officials are the lowest common denominator?
Oh, absolutely not. They're
And I'm kidding, by the way.
absolutely not.
Online.
No, we're all people I guess. Oh goodness, we're all valued colleagues here, but mist from the pulpit is fog in the pews. So.
And yes,
Yeah, I hear what you're saying, and I respect that. But of course, what you've said applies to other taxonomic levels as well. So, obviously, assignment of viruses into species may be important for regulators as well. In fact, often more important than the low species taxonomy. And similarly for family level stuff as well. So, I think there's a wider argument about stakeholders that doesn't just apply to the below species classification that we're talking about here. I'd also like to say as well, so at least for some virus families, the ICD study groups probably take the main role in classification below species. So I'm thinking of the Corniviridae, for example, who maintain a massively complex array of serotypes and so on for the community generally, and communicate it reasonably well. There's obviously Hep C, there's Hep C classification, and many other examples. So I think, there is sort of an infrastructure associated with ICV, which actually may well be able to contribute to this potentially. And obviously that does include, quite a carefully curated set of members and who are very aware of the sort of representation needs to make it effective. So,
yeah, I would say the ICTV study group organization certainly is one possible approach, and certainly even those particular members, as Peter indicates, contribute, currently to some subspecies classification schemes. It does vary a lot between study group as to how active they are. And there's some study groups who are notoriously inactive, as much as subcommittee chairs over the years try to get them to become more active. So it's just a matter of finding new people who are willing. A lot of it's a matter of how much funding goes to one particular virus or not, because the ICTV is covering every virus that it is, whether it infects plants, animals, people, bacteria, whatever. And so the degree of activity varies a lot amongst those different groups. And the level of passion can also vary a lot. But it is still one way of bringing organization to the process of having to maintain the types of information that we're talking about that's needed to, again, define the raw data that needs to go into our phylogenies.
Ryan?
Ryan, NCBI. Something about Elliot's comment just made me think of an issue I think Richard touched on the most, which is the timeliness. So it's sort of related to the maintainability of a system, but it depends on the use case. But if part of the use case is supporting a response to some ongoing outbreak, then there is a timeliness factor that needs to get there, which may run up against
But we may be talking about two separate parts of the system. That is, there's the part that has to establish the procedures, the processes, the type of information ahead of time. That's going to take a bit more time. But then after that, you've also at the same time established a separate infrastructure that acts in the case of an outbreak and is much more timely and already knows generally what needs to be done, and they're the ones who actually go ahead and do it.
Yeah, I don't think it's necessarily a contradiction. I just imagine you could set up a system that just can't move as fast as you want it to, even if you pre-set up the infrastructure. So
maybe the big question here is what kind of infrastructure needs to exist so when the need arises, it's all ready to spin up. And rather than getting in the rabbit hole here, because I think there are examples within ICTV where the reaction time has been slow and examples where it's been fast, right? I think that's fair to say.
Thank you. With respect to the point on communication and stakeholders, I was involved in, still am involved in weekly, now bi-weekly meetings on tracking the evolution of SARS-CoV-2. And when WHO first came along and said, yeah, we're going to call these things alpha and beta and mu, and I at least rolled my eyes, and I remember arguing in those meetings saying, well, we're going to use the Pango names. And yeah, I think a few people agreed, and at least I was pretty sure that that was where we were going. And it didn't take very long until we were all using beta, mu, and we still do today. And somebody said, perhaps, I forget exactly who, that, I mean, part of it is utility and what people use, and also different levels of abstraction. I mean, there's times when we want to know about details of alpha or beta or mu, or, or we want to talk about BA1 or BA2 or BA5, and sometimes you want to talk about the early Omicrons. And so, and having, it sort of gets to the question of abbreviation as well. Do we go with a technical abbreviation that some of which are really easy to remember and roll off the tongue, like XBB, which, a lot of people just say XBB rather than XBB 1.5, which is perhaps the more relevant one and people are talking about. And, JN1, yeah, they stick, but I also wonder whether or not, at some levels of abstraction, it's okay to have more memorable names. It seems less uniform, and maybe just I think that because it's, we sort of do it and we get used to it. And actually, if we never had those Greek letters, we'd be fine still saying B117 and B1351 and etc.
But that variant of concern language really resonated well with a bunch of stakeholders, right? So the public health people, because it addressed their concerns of communication, and others. So
it really did. But us who for whom these numbers completely roll off our tongue and still do today, still talk about it.
Yeah.
I mean, who could be more in this? Well, actually, there are plenty of people here who are more in this. I was going to say, who could be more in this than Betty? But I can now think of the people here that are at least as much. And she was calling them alpha and beta when you did your big sweeps over time.
And moo. Remember moo.
That's right.
Yes.
Exactly.
Your microphone.
Okay. One of the things I heard that I thought was interesting after the WHO stopped with Omicron and didn't go any further was the physician said now it's not evolving anymore.
Yeah.
And I think that's a danger too. When we've got to really communicate better. And this was a prominent physician trying to explain to the public, and so we just have to be careful.
That's a really salient point, because those VOCs were important to a lot of stakeholders until those same stakeholders wanted the pandemic to be over. And then suddenly they didn't want any more VOCs, right?
Can I add to that, though, that having the names changed people's opinions of how they felt about the lineages? So we found when we were classifying them that we had to classify them differently. People cared about why they were not getting a classification of Omicron, why they were not getting a classification of Delta. They wanted to know the specific mutational profile and where it had met it or failed the definition, which isn't the same thing as a lineage definition. It was a different classification for that.
Well, and the classification changed over time, because initially the reason they were called variants of concern, it's because there was something phenotypically in the population, more severe disease, hospitalizations, and all that. And I know in the TAG-VE group with WHO, the discussion was, should we name any of the new variants that emerged, more recently, any of those variants of concern? But they weren't when you look at hospitalizations and disease severity. So they're variants under monitoring, but, that doesn't roll off the tongue either. VUM, who cares? But so that's the problem, right? So because the pandemic has changed in the population, now we're sort of looking at it in a complete different way. Cornelius.
Yeah, I think the stopping with Omicron problem shows that it's tricky if you do things at a phenotypic level. I mean, let's imagine there was no Pango and no next strain clades, then you would have nothing left to talk about, right? Yeah. So it's maybe you need that other level, or that other level is useful once you start talking to a broad audience and once the public cares, but it doesn't mean that you can't do away with a genotypical quite low level system.
I think that's what you were saying, Richard, right? When you said the genotype is very important, and then you just relate your phenotype. I wanted it somewhere here. Do you even remember what you said? Positive selection. Use your mic.
Yeah, positive selection.
Yes.
But so another thing I just want to put on the table is that, so one aspect of this is to try to understand genomic evolution. And I think Betty brought this up in your presentation, where, we're really good at tracking nucleotide substitutions over time. But then when you have that recombination event, now all of a sudden we still throw these things in the same phylogenetic tree, even though we know that it completely violates kind of, the topology. So I think we also need to be thinking about these other forms of evolution, like recombination, genetic gains and losses, gene gains and losses, and reassortment events. And we don't really have a good framework for tracking the lineages of those things as they're emerging and evolving.
That's almost like another implication. That's almost like another question here, which is, what's the right way of consuming this information at any level? So we look at the n-dimensional sort of layers and layers and layers. Are trees the best way of viewing that kind of information for all the viruses that are in scope in this conversation today? or does there need to be something else? And then the other thing I was saying, going back to Betty's comment, if you're going to have a nomenclature, it's got to consistently be applied, just to underscore it, just to make sure we capture that. Because, if we have like a nomenclature that is going to be communicated to people that are non-scientists, like neighborhood, then it can't just go away 1 year. It has to be maintained. Because otherwise, 5 years from now, when neighborhoods come important again, all that infrastructure of maintenance has gone away, and now you have to resuscitate the system. Because
Can I add a caveat that disagrees with that? It has to serve a purpose as well.
Yeah.
And if the purpose is gone, then perhaps we should let it go.
Actually, agreed there 100%. Let things go by.
I agree with that too. But I also think that then it's important to say why we're letting it go and have that communicated really effectively. And maybe that's what was lacking when they stopped at Omicron.
Because
Hey, this is Anita Hatcher from NCBI. So responding, I think, to Rodney's first question there, I think one of the fundamentals that we should be thinking about is how do the different methods of evolution in different viral groups play into the decisions of what kind of schemes are most acceptable. So for some viruses, a phylogenetic tree-based like evolutionary system that tracks hierarchy is going to be perfectly suitable. For others, it's not. So maybe that's one of the dimensions when we're thinking of how different schemes can apply. One of the dimensions might be, what are the evolutionary methods favored by these viral groups?
Well, I agree. And again, that depends a bit on the level at which you're trying to classify things as well. So obviously, this phylogenetic stuff's great for SARS-CoV-2, but it's no use for Hep C genotypes, which actually would be defined usually by similarity thresholds, which is obviously quite a different approach. But also, just like to say as well, that I would imagine the classification is permanent. I don't think we just disband them when the virus becomes less important clinically. I think that's completely wrong approach, and I think we need proper ways to record classifications so that actually it can survive beyond the enthusiasm of the people who did it originally. Because it's the basis of the rest of taxonomy.
We have a couple online.
So there are three different comments. The first one is related to designing the classification system to categorize viral diversity is essential, but what's the surveillance mechanism and how frequent these are for the different viruses? If you're not surveying, that means you're not tracking it, the evolution of those viruses. So what's the correct frequency based on the virus that are circulating in the population?
Yeah, I would say in reference to that, something that we really haven't talked that much about yet, if at all, really is what role does sampling bias have in coloring our perception of what's going on?
And when you said, sorry, the question, when they say frequency, it's frequency not of whatever is circulating, it's the surveillance.
Surveillance and sequencing, correct. Yes.
So that's another whole infrastructure. That obviously we know needs to exist. Because we all work on data, but obviously the people at the front lines collecting the samples and then doing the sequencing are critical to generating that data, and we can't do what we do without it.
And I think that goes back to something Betty said at the beginning about areas having access to even getting the sequencing. So I worry that we'll have this incredible data and incredible structure for half of the world, and then the rest of it is very limited. So we haven't talked about that much.
There's another comment here related to the phylogenetic tree or the visualization. Tree might be purely based on the sequence similarity, which is true, which might not be robust to show the information about the recombination, reassortment. So this brings it back to all the dimension data that we want to track. because we are touching the different dimensions that we want to track. Because the people who are tracking the pandemic, some people are interested in origination of the pandemic, and some people are, from the health perspective, wants to respond to those pandemic from the health perspective. So the different dimension in all these are important for how they are responding and where they are responding. So these dimensions are very important, and we need to categorize those dimensions. Because
Oops, sorry.
I think I said this earlier in the day, but I guess I just want to emphasize it. When the SARS-CoV-2 pandemic hit and we got the vaccine within a year, millions and millions of lives were saved. There's a beautiful modeling study out of Japan that really brought that home to me. It's fairly recent, but it really made a huge difference. And I think serving the immunology community with nomenclature is really important. And they're not all phylogenists, and they're not all, sequence jocks. They do something different. And I think in terms of saving lives in the next pandemic, they're going to be at the front lines again, just like they were last time. And what could have been had we not had that vaccine in a year is even harder to imagine. So anyway, I think they're a particularly important stakeholder, and they depend on the nomenclature.
But do they care about the format of the particular name, or just that there's a unique name to a lineage that may have a unique antigenic profile?
They certainly care about the names a lot, and I don't know. But, the names are very important to them, and it's their language too, but they don't always necessarily. So I think reflecting immunology somehow and being able to tie back to it, as well as the phylogeny, which we're all very, up on, is an important thing to remember. I think they're maybe the most important frontline community that we've got, at least in the current world of science. That may change, but
isn't why the original subtyping was based on serology?
Yeah, and also we weren't so good at sequencing. We've gotten a bit better, but both of those things are true. Yes.
But Betty, that point notwithstanding, to the heart of your point, it seems to me, no matter how you do it, no matter what the structure of creating a genotyping scheme or a subspecies classification scheme or a naming scheme, I think that it's key to know your stakeholders and know what each stakeholder needs to know from the scheme. I think that's a key component to whoever undertakes this, and at whatever level of, generality you try to make it.
Which ones to test? That's what they need to know. So, yeah.
Because questions more like JCVI and BV-BRC. Yes, questions more like JCVI and BV-BRC. I just have another general comment. Maybe it's not even appropriate for this session, but a lot of these classification schemes, they're based on some sort of reference sequences. Because, not all of them, I would assume most of them, and how those reference sequences are selected, and how big your set is, and especially that they cover whatever you want to cover. I think it's also very important, and because it influences the accuracy of the resulting tools which you build on this based on reference sequences. I think that's also something, maybe a topic even for another workshop, but it's also very important.
Because I think that can also help address the sampling bias issue. If you have a way of selecting representative sequences, you can also use that as a way of controlling for the ascertainment bias.
I think it's back to the point Betty was making earlier this morning, that because to have good representation, you have to have more sequencing going on around the world than is currently going on. And I think that's a key thing. But I got in a conversation, to your point, with someone earlier, that a lot of the schemes we looked at today are reference-based, but there are schemes that are less dependent on references, where you create essentially a model, and that model becomes the reference for analysis. So,
it's true. Philippe from Swiss Institute of Bioinformatics. So that's something we have identified very early, that
when a new variant came out for SARS-CoV-2, for example, there was a lot of advertisement. You got some mutation profile, but no reference sequence. And many people need for the study to get a sequence, actually, or they will not type manually from the mutation profile that for Omicron was very long, for example. So we tried to provide this, but I think it's very important that at the end of the day, different groups have provided the GCVI. We did in Viral Zone and other people in CBISO. But at the end of the day, we should prepare that when a new variant came out, that not just getting a mutation profile, but a reference sequence. So after months, we agreed together to agree on the same reference sequence, and it was afterward, and it was pretty nice that we did that. But users were already missing to get on the point a reference sequence when we said there is a new variant, because many people making studies are really people mutation profiles.
Actually, that's something we do in the SAVE program. We established that we need to put in a number, a GISAID number or a GenBank number, so they can pull it out. But there's another point that's really important with that one, and it's that when a new variant is coming out, there's all kinds of sequence errors because the primers start failing. So at the time everybody wants it, when it's just emerging, there's lots of ends in the DNA sequence. There's also bioinformatic mistakes that are happening a lot, a huge amount, especially in the first 2 years of the pandemic. We've gotten a bit better at it, but people would put an N as ancestral if that was the dominant base in a set. So almost every single new variant would have a lot of N's in it in key places, the most variable places. And then we call them ancestral. And so over and over again, I kept having to correct people who would lose $50,000 in a month or two of work because they were making this thing that had ancestral amino acids because they didn't actually look at the alignment and say, oh my gosh, in these 500 new sequences that we've got out of Omicron, everyone that has sequence in that region has this sequence. But the 70% that have N's in that region are getting called as ancestral. We're still having those problems a little bit. And then these things get out in the world and replicated. The other thing is people weren't putting the sequence they were using in the experimental papers, getting different results in terms of the pathogenicity, antigenicity. So if you don't have this actual strain you're using for the phenotyping, you can have those mistakes. And these things were really very, very common mistakes throughout the pandemic and a serious problem. And I think people didn't understand the importance of these factors. And so we tried to establish in the SAVE project some very strict guidelines about knowing that it was the spreading strain and having an accession number that could point to the consensus from the strain, because they're all evolving all the time, right? So you want to catch the thing that's spreading, the central, most common thing that's spreading, and give that to the virologist and immunologist to work with. But it's not always easy to get that. Indels also caused a lot of problems for people in the pandemic where they would get the indels wrong.
It's a good point. I mean, maybe a consensus would be better than choosing one sequence. Whatever it needs to be thought about.
For SARS-CoV-2, I would say that's not true because as you had a newly emergent strain, there was huge amounts of identity with lots of N's in them. So the consensus would match 90%, 95% of the actual sequence that got through.
Yeah.
So the consensus and 90% of the spreading variants in the new variant would be exactly identical. So, that's why it was nice to be able to go to an accession number of an actual viable sequence. And in fact, our other components, our in vitro and in vivo, did not want a consensus. They wanted an actual sequence. They wanted a real bona fide sequence. So they said that repeatedly to us in the beginning. So just going back and forth.
And before we move on, just wanted to also, I know for the NIH, Liliana, correct me if I'm wrong, but for the in vivo, in vitro work, we can't synthesize a whole virus from a consensus. You have to use a real virus, right?
People,
Because you can't do mutations in a real virus.
people.
It's like
the gain of function
issue. I know there's like something about.
So it's always simpler to get a circulating isolate for many reasons than synthesizing some. Most of the work that we have done here has been using just the spike protein. So the coding sequence for that is usually synthesized or site-directed mutagenesis or something to that effect. I think we found that getting the isolates, at least during the pandemic, was faster than trying to do the synthesis just because it takes so long to generate synthetic constructs. But at the very beginning, where there was a very limited number of isolates, certainly we had to go the pseudovirus way or trying to make synthetic constructs. There are, of course, waivers and exceptions for doing this type of work, especially related to in a pandemic situation. So those are things that are evaluated and monitored, and there is research oversight. So it's not completely prohibited.
Thank you, Rihanna. First, Justin, and then I'll go to you.
So I think this idea of having a reference strain is really important, right? I really value that, right? It allows our collaborators that are trying to pre-pandemic preparedness and select vaccine strains, they have something to point at. To be devil's advocate, how can we assign a lineage a name when the virus sequence is not complete? Right? Like if that may not be valid as soon as you get that complete sequence because you have a bunch of ends, then it almost seems like it's a moot point that we're assigning a name without a complete one.
So I think Cornelius can answer that because there has been changes adapting the names.
Or at the very least allow it to be corrected, right? That you could sit down and say, this is the putative type pending correction. At least then people can sit down and say, all right, there's a type sequence, but it's being completed.
Is defined by a number of designated sequences, not a single sequence. So in the Pango system for COVID, it's defined by a number of sequences, a number of designated sequences.
And you can reconstruct a founder that kind of is the common ancestor of all those sequences.
But the point is that you for partners, they can't use a reconstructed ancestral sequence, right? So they can't use a reconstructed ancestral sequence. And if it's based on
why can't they?
I thought that was part of the issues that Elodie just raised.
Prefer not to, but
so actually though, with any of these new, in SARS-CoV-2, with any of the new lineages, so what winds up happening is you have these little stretches of ends. So you'll have it's beginning to expand, and they're very related when they're beginning to expand. They're different than what you've had before. So the primers start to fail a little bit, and those have to be tweaked, and that may take 4 months or something. So you have these fragments that work in some labs that don't work in other labs. This is the reality of it. So you don't always have a majority of the sequences that are spanning that region, but it's Omicron. So you know that what Omicron is like, and then maybe in 60% of your Omicrons, you're missing this little bit, but the other 40% have it. And so you can see what should be there if you look at the alignment. It's early, so they're really close. So the ancestral form equals the consensus equals a lot of the viruses in that first set. So that's what we would always pick out and give that. And it would equal the ancestor of that clade. It would equal at least the spreading ancestor of that clade. It would also equal consensus ancestor and, a good fraction of them. It's just that what was getting put on websites and getting put in tweets and getting put in papers would be something that would have reconstructed the ancestor where these ends were. And they would be a false sequence that actually didn't exist in nature.
So they would have reconstructed the Wuhan who won as opposed to actually the
So it was actually the real form that was yes. And that happened many times and cost a lot of money and a lot of wasted work. I think a lot of ambiguity in the early experimental. And I watched it happen. I know this happened a lot.
the reason why you would want real strains is because they're easy. There's kind of no human inference step involved, right? If someone reconstructs a common ancestor, that involves some computation. Whereas if you say, "Oh, I'm sequenced that sample, this is what the sequence is," there's less deduction involved.
There is also the gain of function concern. So if you're just looking at spike and you're doing a pseudovirus, you can do what you want. But if you're looking at a whole virus that you're infecting animals with, you have to be more cautious. And I think Lillian and Elodie both alluded to this, but there you don't want to create something artificial because you've got the risk of creating something more dangerous. So you need it to be an actual strain to do the animal studies to determine if it's more pathogenic, if it's more contagious. So what I do is they'll get a real strain, and almost all the real strains somewhere in that 30,000 bases have a couple of changes. So figure out where they are, look at the consensus and say, this one's got to change, in or fate. And then let some experimentalists know that the one they got, the one Liliana's found to share, is mostly normal, but it's got these two changes. And so we compare, I think ancestral or consensus are right there. And that's what we've done throughout the SAFE project. So we're kind of we're there as a resource for the experimentalists when we got this new isolate. I'm sorry, I'm taking so much time on these issues, but they're ones we've kind of hashed out over the years.
Yeah. So I think Christian had a question first, and then I'll go to you.
Would be a question more. It's actually two comments. I mean, the first thing is the ancestral sequence reconstruction itself is tricky or complicated, and especially if they're dealing with very short distances. You cannot. There's always multiple solutions from the ancestral sequences. So you not only have one sequence, you have many sequences because there's uncertainty how to predict it. So that's my first comment. The other comment is, I mean, I didn't even think I need to say it, but when I was talking about, having reference sequences, of course, the first step should be like quality control, like expected length, and of course there should not be sequences with ends in there, especially like runs of whole ends. I mean, that's it's kind of common sense. So here, that should be obvious.
So here the ends is not just the quality control where there shouldn't be ends. It's the fact that ends were interpreted as being the ancestral state. So that's the quality control. Yeah. But, we think it's obvious, but that's when, you see people doing all this weird stuff. And you're like, why are you doing that? It's because in whatever their training on the genome they worked on, I talked to a yeast biologist who was doing that automatically because that's what they do with yeast evolution, but we don't do that in viral evolution. But don't assume anything, basically. Yes. Ryan.
Hello. Ryan, NCBI. I think all this discussion, I'm thinking about how it connects back to subspecies grouping classification scheme and subspecies. They meet up perfectly if you come up with a new classification for every single mutation that occurs. But the further you get away from that, more, it's not necessarily a problem, but there needs to be, subs being intentional about how you deal with this. Because if your groupings are very large, maybe not every experimentalist wants to work with the same single reference sequence for that large subspecific group. But then they probably still need some way to talk about that. And I don't know. It seems you're trying to come up with a somewhat general solution to the classification problem. That seems like a issue that needs to be grappled with.
But there was another question from
subspecies. I wanted to add when we were talking about the variant references that generally they were updated very quickly in a short space of time because there was such scrutiny on those sequences. So, Omicron, the reference was updated overnight multiple times for the sequences that we did have. And this meant that the overall sequence we had was improving every day, but we knew which sequence it was. And it meant that we're talking probably a timescale of a week, maybe 2 weeks between identifying. Yes, it's long, but it's still it's long in pandemic times.
Yes.
It's short in other time scales. So,
exactly,
how quickly do we need a specific reference?
subspecies.
I don't know if this is what you're getting at, but what we observed is when you had a dramatically new lineage emerge, it very rapidly evolved because there were some fitness disadvantages, when it initially came out. And so then it stabilized after a couple of weeks or something like that. So if you pick the reference right as it was emerging, that was not really the thing that ended up spreading and circulating. So, we kind of felt like we should delay just a little bit before we selected a reference.
Yeah, how many times has it happened where on a Monday we're talking about a lineage, and then when we have to give our rundown on the Friday, we've already changed the lineage we're talking about. Yeah.
We have an online. Yeah, this is related to the ambiguous base or the gaps.
Subspecies.
So the question is gaps of large number of unidentified nucleotide mutations by WGS and their respective actual biological functions confirmed in the lab. So if there are gaps, those should be closed?
Subspecies.
That is.
Subspecies.
So closed by follow-up sequencing?
Correct. Yeah. So you don't want to leave those, especially when you're using those as a reference for a strain.
Subspecies.
Right.
There were not not all labs had the capability of doing that, right? A lot of these emerging variants came from labs that just didn't have those capabilities.
The samples may not have been up for it, right? Depending on the samples for target.
Sure.
Yeah, I was just gonna, kind of say the same thing. I mean, many of these samples were sequenced not for the purpose of generating the perfect reference, right? Because they were sort of at the borderline CT value, and just you got what you got. And yes, the ends actually aren't the problem. The problems are sort of contamination by other variants that filled in these sort of low coverage regions, sort of dropout regions. But then you have like 5 delta reads that come in from a sample that you did 2 weeks ago, and generate these delta cron chimeras. So that was the real problem. So if somebody calls something an N in the sequence, that's thumbs up, right?
Problem there, though, is that a lot of bioinformaticians had code that called the N ancestral. So it wasn't.
But that's exactly what I said, right? So if there were N's in these places, that's good.
That's. I get what you're saying, but I think I'm not making my point clear enough, and I'm so sorry. I know I'm lingering it.
We got it.
Yeah.
These are different problems, right? There's both the problem of bioinformatically assigning a nucleotide when you should just leave it at N.
Yes.
And then there are 2 problems. Sequencing problem where when you have very low viral titers, so low coverage, and there's a gap in a region, any contaminate in the same sequence. One of the issues is that in high throughput sequencing, we're sequencing in real time, right? So you have all kinds of diversity. Some things are covered at high coverage and will contaminate your low coverage, parts, and then create a chimeric sequence where the consensus is all closed, but it's wrong.
But I feel like many of these issues are more like, standard operating procedure problems and bioinformatic problems, and not so much subspecies classification.
No.
That's right.
That's part of how do you choose your reference, which we use for classification, Ryan.
This is less a classification issue and more just a general bioinformatics thing, but to the extent sequencing is becoming more common and done by different people, and I don't know, maybe it's good to be more explicit and not assume everyone had the same background training, but for SARS-2, comparatively speaking, it's easy to make a consensus sequence out of. You can do just align to reference and call consensus. That is, in my opinion, not acceptable for a lot of other things, and you need to do a much more intensive process. But saying when you need to do what process and like having a way to track how you went about doing your consensus, so someone who has a strong opinion and doesn't like your method can exclude it, I think is an important factor in for having a widely shareable, usable sort of standard.
Justin?
I wonder if a little bit we're focused on the exception for SARS-CoV-2 than what should be the rule for moving forward. Because SARS-CoV-2, of course, it's driving a lot of the conversation, but we're talking about applying standards across the field and how we communicate viral evolution.
Good point, Justin.
Question from Zoom. This is coming from Ellison from HHS. For the pandemic preparedness perspective, major concerns are that names are granular enough to communicate difference between the phenotype of vaccine and the therapeutics. Doesn't matter what you call it. And she agrees with the Betty's comment about the immunologist being able to access that in the lab. So it's important.
Oh, yeah. Perhaps we are hearing too much about SARS-CoV-2, but maybe not. Maybe there are really two different paths. One for, a huge outbreak where we get thousands and thousands or millions of similar sequences like SARS-CoV-2, and another for other viruses that have already been diverging forever. Those are two really different problems, I think, two different use cases. So we might want to keep them.
And that takes us back to Elliot's point, which was the fact that we need to have some kind of robust system that you basically have a structure so you can use the similar structure, right? You were saying, Elliot, you use a similar structure that you can already have a process when something new emerges, but that you could apply for what is already endemic.
And I would envision a series of SOPs that would define that structure for the people actually doing the work. And concomitant with that, there may be need for a similar series of SOPs that drive the generation of the data in the first place. I mean, individual labs have that, but I don't know that there's anything that's utilized across, all public health laboratories for sequencing, for example.
Anya Till, University of Edinburgh. I think an example of where that has been, or when it's been proposed to be the case, is with MPXV, where you have the clade resolution system. And I think for MPXV, that seems to be, an appropriate level resolution. And then when there was the human outbreak, you get that finer scale resolution and it gets introduced at that point. You're not trying to characterize all of the deeper diversity with a fine scale system. And then say, something else comes along and you want to start that system again. You can adopt that finer scale resolution when the question presents itself that you want to answer with a fine scale resolution. But having this sort of broader stroke clade system where that kind of characterizes the deeper diversity in combination with the fine scale when appropriate, I think is important.
It also helps because something, that was important for MPOX as that the clade nomenclature was developed, was that you had long-term pox virologists sitting in the room, sitting on the WHO call, along with the new people developing the very fine-grained lineages that had been used for SARS-CoV-2. And so bringing those communities together to derive a system that everybody was in favor of was an important aspect of getting that agreed on and making it work.
Ryan again. This is more of a question, but so a lot of the talks we heard from are pretty well-studied viruses that all have classification systems in some state of quality. But some of the discussion, particularly thinking about like pandemic preparedness, is maybe for things that aren't so well studied. There aren't that many sequences. And I guess the first, it's sort of like a basic, almost like dumb question, is just, when do you have enough data? Because in theory, it's nice to say make a system that works for literally everything in the abstract, but I don't know, that seems unrealistic to me personally. So where do you draw the cutoff to say this is worth actually thinking through? We have enough data, there's enough cause to be interested to come up with and create the infrastructure for a system. For just harmonize all the existing things that are well studied, or is it also do other things that aren't so well studied yet?
It's the kind of thing you know when you see it, right?
Yeah, it seems like we need to come up with classes of viruses. So for this class of virus, you take this strategy. For this class, you take this strategy. And there's probably not one size that fits all, but maybe there's 4 or 5.
Yeah, I wonder though, when you start making sort of rules like that, and then you go down this rabbit hole because there's always the exception, you know? And so I wonder if we keep it a little bit of here are all, that's why I keep on going back to Tavis's different dimensions, because you have all this kind of information and you can just pull it when you need it, right? In your classification. 3 minutes. All right. So Tavis, maybe you can close us out.
Oh, hopefully not close us out. But I was just thinking about
closing us.
Oh, I am closing us out. Okay. I better be good then. Just thinking about undiscovered viruses and undiscovered diversity within classification schemes. And there are ways of quantifying uncertainty with predictive modeling. So that could become a metric used in terms of these classification schemes so that you quantify uncertainty due to ambiguity of the data, and then quantify the uncertainty due to the model's familiarity with your training data as well. And you split those apart so that then classification becomes not ambiguous, but you can quantify uncertainty in your classifications. And that could help address the unresolved issues in classification and unseen viruses. So there may be areas of the landscape where we just haven't sampled yet, we haven't surveilled. But then there are other areas of the landscape where we have conclusive evidence that these are the viruses that we see here. So quantifying uncertainty in our sampling. Thanks.
All right. Well, we can finish this at the bar. We were not trying to solve world peace today. We were just trying to, this seems a brainstorming exercise. And I guess, as the days progress, by day 3, it'll be all clear.